Okay, so the next thing we're going to do is actually install FastQC. So you're each going to install it in your own Scratch folders. And then we're going to just run it and see the results. You're going to download the HTML file that the software produces with these plots. One thing I wanted to mention is that for convenience, because all this is, we're going to start running things so that things take a little bit less time, and I should share the different screen. That's not it. This is the workshop directory, the workshop folder on the cluster, the main one, which you don't see for some reason. Oh, I need to do that. Training post. Resume share. Okay. So you couldn't see it, but and on the right. Right. So I'm on the cluster here, and then this is a QCBW5A directory which we have used last time. And then I've added these two files. One's red and one's green. Anyway, the FastQ, so the top one is the one we're going to use for the beginning today. And then later on, we're going to use the BAM file. And we're going to go over BAM files. But as the names of the files say, these are just downscale from the original files, which you have manipulated yesterday, to 110. Okay, so everything's going to be a bit quicker. If you have to download them, we're going to download. It's just going to take less time. So yeah, these are new files. Copy them to your Scratch directories. Yes. And so the first thing I wanted to go back to before we actually start with FastQC is this question regarding, I can't see. FastQ files. So the question was, if we take back the FastQ exercises, was, are the qualities of the second read good? So I'm not sure where my mouse is. There it is. So this is this file for the first sample here. So I'm just viewing this. And so this is, and the question was about the second read, just because it was less ambiguous. So this is a second read. If I make it a bit shorter, we can see the end of the read. Yeah, so these 4 lines, lines 5 to 9, there's a second read, right? 4 lines per read. And then the fourth line of the read is the one with the qualities, right? And so the qualities we see, we have mostly I's and then H's and some F's and G's. Mostly, yeah, there's one D there, but so we have these letters. So what we're going to do to, what's going to give us an idea of what sort of qualities the read has is going back to the table that I showed yesterday. Where's, I need to zoom, yeah, this. I can't see it anymore. Oh, it's gone. It's gone. I don't see my mouse. It's a bit complicated. That one. Okay. So this is, okay, so if I go back to this, so these are the exercises, and just before that we saw this. So these was the slides about qualities. And what we can see is that all of the capital letters are actually at the very end of the score scale, meaning they're very common. They're between basically 35 and 40. So all of these bases from the second read, they were actually very good. But basically what I mean here is that if you have a FastQ file and then you look at the qualities of your FastQ files, and sometimes some clusters will, some sequencing core will just send you FastQ files. All of the first reads are just very bad quality. just because of the way they were sorted. And you're going to be able to see, if you see lots of punctuation mark, usually it's not great. If you see lots of capital letters, the reads are good. So yeah, that was all like looking at what characters equality were, and then looking up the characters in this table here, which is going to be about 30, so they're good. Okay, so yeah, let's switch to running FastQC. Okay. Yeah. Oh yeah, and the goal for today is to do mostly just practice. I'm not sure what you mean.
Here, I cannot see those files.
Oh, it's because there was an error there. Oh, it's because your DAW scratch, that's going to be your scratch. In my case, it's going to be my scratch.
Yeah.
So there are different folders. There's like your scratch and my scratch.
Yeah.
Oh, so try. I mean, first of all, what you want to probably is this. So this exists, right? And then I'm not sure what you have here. Was this copy of data workshop file?
Yeah, this is
So
what if I want to analyze the data here? I can use my own data.
So can you open the Hoffman 2 PDF for in the Google Drive? That one is like the last page, I think. No, this one's blank. Oh no, previous to last. Oh, I don't have it. Okay, I had not realized they were slightly different. And so just to remind you, this is where the workshop data is. This is a full path for the workshop data. This slide is slightly different from the one on the Hoffman 2 PDF. So this is currently where you are. So let's do this. If you want to give it the best way. Or you can just replace the Oh, no, that's a web page. We're actually downloading the. And you can just copy. And you have basically just generally. I'm going to microphone. Generally, when you want to download programs or files from the internet to the cluster, you have two ways. You can download them directly by clicking the link on Internet Explorer, Firefox, Safari, whatever you use. And then that's going to be downloaded to your laptop, and then you can upload that to the cluster with SCP, for instance. The other way you have is you copy the address to the clipboard. So usually it's like right-click and then copy link address. And then you paste this into, while you're all connected to the cluster, and you do wget on the cluster, and it's going to download that from that address directly to the cluster. That's usually a bit shorter. It's the same. But yeah, you always have the two options. Whichever is more comfortable and easy for you. I'll show in a minute. I mean, the goal is really practice. So if it's a bit, if you need to think and try to make it work, that's what is intended. And this is on the cluster? Yes. Yes.
And then I thought now I okay, oh my god, but when I use
I can go back to FastQC installs. Yes.
so I downloaded FastQC.
Oh, so what's indicated here is the way where you directly download to the cluster. So you doubly get the address of the file you want to download. Well, it has to be a file. It can't be. It's not going to be a web. I mean, it's going to be a web address, but it's not going to be the address of the website. It's going to be the address of the file. So in your case, you don't have to do wget.
I don't have to use wget.
No, because you've already downloaded the file.
Okay.
Oh, wget is for webget, get from the web or whatever you want. And unzip is a bit like gzip, but for .zip file.
So I did the wget twice.
Oh no, actually, if you do wget on the website.
Oh, do you want the website?
You're doing wget on the website. So what you're just loading on the website. I mean, the website itself, like the HTML page. If you go to the website, oh no, just copy paste the address here. Well, if you click directly, you have copy link, and if you go to the terminal and paste that. So just paste it. I need to well, the thing is, the address. download. Oops. Oh no, you have to click the link.
You have to click the link?
And then that takes you to a different web page, and then you can download.
And then copy paste that.
Yes. And it should be the link you're copying into the terminal should have a .zip at the end. It tells you the file does not exist. So that's kind of why you want to always use documentation, is because basically the file is not where you think it is, right? This is where you are. So if you go to one directory and you do so you are not in the right directory. You're trying to use the computer we're thinking there should be a file in this file should be in the other. If you have to get the right exact address where the file is, otherwise the Linux file. The Linux release. Yes, you're downloading .html, that's not right. So Uber or the FastQC version 11.8 Windows Linux.
Yeah.
So Uber or the no, just the Uber is the change the focus to your browser window, and then copy the link, and then copy the link or the link. The presentation, the link. You want to use Control-C keyboard so that you have the full screen. Just do and they will get face. Can you copy that again? If you copy to the try Control-Shift. If only in Turing. It's something with it's actually something with your terminal window rather than it's not anything you're doing with the command line or anything. It's something with the window that this man wants to. So in your case, it might be easier to just download it to your computer and then send it to you. So it doesn't work for any of the commands we've typed in the past. So usually I'm trying to not have to just copy paste from the PDF. That's fine, but there must be a way. If you look it up on the internet, you might eventually find a way to copy paste. That's because actually, so you want to download the Linux version. Those of you who have Macs want to download the Linux version and then just upload. If you're downloading, even if you're downloading to your computer, want to then upload it to the cluster directly. It's just a different version of compression, whether it's a PNG or how the zip file. It's just the compression that the contents is actually the same. That's cool stuff. You don't change it. Yeah. Just in this case, just calling Windows push the Java website. But when you use Java for FastQC, you might need it for and generally, if you're going to run like some software, it's a good idea to have Java installed. So instead of solution separately, and then copy this. It's just like the same file. The archive format's different. So the contents goes to the same file. Then you should have the file in the laptop. And then you can STQ the file to the project. And then install it. Oh, that's different. That's just because it's trying to download the file to your current directory, and you're putting still in my scratch directory, which you don't have permission to write to because they're my directories. So everyone can only write to their own directories in general. So it's telling you you're trying to create a new file in this directory, but you don't have permission to write. That's it. That is right. So just change your directory to your own scratch. Alice? It should have worked. Not yet. Not it's. I mean, the terminal is kind of hotspot. Now it's green, and that's because you can run it directly. So if you try to just run it, just type FastQC. Oh, it actually is going to be /FastQC.
FastQC.
No. So just type / no space, and then FA. Yeah, tab. Yeah, like this. Okay, it's trying to open -H. Try the same thing. Oh, no, sorry. Just reuse the previous command. Control C to remove these characters. Just up arrow, the arrow like the actual arrow line, like right up. Yeah, and -H. Like help. No, just dash up, and the H is for help. Okay, enter. And then you have the help for FastQC. And if your terminal is a bit bigger, okay. Yeah. It's because that's the path variable is quite a variable. What the path that you should have put it there is actually FastQC.
Oh, I see. I tried to.
Otherwise, you can directly do the path command like stop and store / FastQC / that would also, but then you not have to use the path variable. But using the path variable. Yeah, it's running that. Yeah, so actually, If whenever you see that the fast has become green, it means that you have successfully made it. And then it's just about running it.
Yeah. So yeah, but then it goes on.
But it's the same, but it's not changing.
Oh, yeah, I'm not sure, but I think I might. So fine. No, I think, yeah. I might have tried to I read my hands.
So, well, by the way, in the chmod command, the U means user, that means yourself, and plus X means add the X executable permission. Yeah. And then, I think it is.
Okay, so I need to be in the. Oh, okay.
You see Java errors, it means it's trying or X display is trying to open windows on the cluster, which it cannot do.
Is it just jumping? Like the error? What is the dash issue?
Okay, so once you have run FastQC, it should have produced for your FastQ file, it should have produced an HTML file. So download that HTML file to your computer, and then open it with, I mean, you can double click it. It's going to be open in Safari, and you're going to have these plots that we've shown. And then we'll switch to the So actually just FastQC. Yeah. Okay. I'll just show the. Basically, just to go over this exercise. So going back here, and then where's my? How do I take this away? Move this. Okay, much better. And new share. Okay, so I'm here, and then so I have my. Okay, just going to. Demo, can't see anything. So demo, cd demo. Okay, so I have my zip file, unzip the file. Okay, and then I can just cd there, chmod u plus x FastQC. Okay, so now I've got this file, which was not executable before, has become executable. And then the other way we can look at this is if we do ls with the long format with -l, and then this is our FastQC file, and then there's this x here that has appeared, which is not on the other files. Okay, and directories are always executable. That means actually you can just go into that directory for directories kind of a different meaning. Okay, so to run FastQC, so I'm here, and then I'm actually going to copy this file here. Okay, so I've got my file, my FastQ file here that I want to process with FastQC. And then the FastQC, many of you have noticed if you just do FastQC, oh, actually in my case it works. But normally it would not work. It works because it's installed elsewhere in my directories. You actually need to do that. So this command here at the top, which has not totally disappeared yet. And the problem is when you just run it like this, it's actually trying to open Windows on the cluster and everything, and then the cluster complains, and then FastQC produces errors. Okay, that's what's happening here. So it's kind of normal. Basically, what you want to do is -h or --help. That's the same. When you have two dashes like this, it means the long version of an option, and one dash means a single letter option. So you can do -l like this, and then it prints the enter help. And basically, in this section here, you can see that basically usage is, you can use it on multiple files at the same time. It's not necessarily what you want to do, but you could. So it's just FastQC sequence file. We're just going to do that. And again, we need to give the address for the program because it's not completely installed like to all users and freely callable. And then just our, and here what I did was a stab thing where I've got multiple files, but there's only one starting with a P. So I write P and then tab, and it just gives me the file. And it's still complaining. Oh, I'm sorry. Actually, yeah, the gzip thing is my fault. Okay, I see what the problem was. Sorry about that. Where's my mouse? Let's rename the file. That's why it took a while to come through. So it was .fastq.gz, but it was not gzipped, and that's why FastQC complained. I should have checked that when you told me. Okay, it's going to zip the file. I'm just going to let it do that in the background, and going back to this demo directory, and then I'm just going to wait for the command to, actually, we're just going to wait for the gzip to finish. May take a little bit. And so then we're just, basically that command, this is basically what you were expected to do, okay? Okay, so it's done. And we can just do that. What is it still not in zip format? It's because it's a copy. I see. Okay, so that's basically FastQC running. So it's doing this running for like 30 seconds, and then so originally we have these these two files for this directory in the current directory. And then when it's going to finish, not too sure what it's doing now, but it is. Okay. Okay, so we have this new file here, and this one. And so this is a file that we want to download to your. So we're in this directory. So if I exit my session on the cluster, now I'm on my computer, my laptop, my MacBook. And then I can do scp my username at auth dot, and then the path to the file, which is this, and then so directory, and then the file itself, and then I just copy here, right? And then if I do ls, it's going to be here, and I can on Mac you can do this like this. It's actually open on my Firefox, which I'm going to try to share now. Where is this? Why is this? Come back here. No. Oh, I still need to do that again. Okay, this is my Firefox. It's just struggling a bit with the Zoom meeting. Okay, so this is the file that we just created, right? This is the HTML file, and we have these plots that we showed in the PDF, where we see the base quality, and then we have one peak and one fairly high peak for FASTQ score with everything almost above 30. And this is the per base sequence content, GC distribution, and different things. And yeah, this is what I meant. I can zoom out a bit. So this is the sequence duplicate levels. And one thing you can see that there are quite a few sequences that are duplicated more than 10 times, but that's just transcript being sequenced multiple times, just because some transcripts are more abundant. Okay, so this is it for FastQC. Let's switch to Trinomatic. Okay, so read filtering. Basically, this other software is just so a number of mostly reads are good and the run just looks good. And the next thing we're going to want to do is not just throw all the reads into the alignment, just because some of the reads are not as good. Some of the reads are always bad. There are just a few reads that are always bad. So we just want to remove those. And then some reads, something happened, and then the average for some read qualities just dropped for some reason after a number of bases. So we're just going to remove the bad reads and then trim the reads when they quickly drop in quality. So just remove the bad bases, remove the bad reads. And we're going to do that with Trinomatic. I might just show you how to do it, and then we can switch to the other PDF. Whoops. Okay, so I can just go to this address in Firefox. Oh, I can't select. So let's just go to Firefox. I can switch like that. Okay. I can just look for Trim-O-Matic. And then where's it? Download. So I do copy link location, and then going back to the cluster. And okay, I have the link. And I can doubly get that. That's not the cluster. No, it's not it. Just doubly get that, and then unzip it. Okay. And then I've got this .jar file, which is a Java file. So I want to do module load Java, then Java -jar Trim-O-Matic. Oh, it needs that. It's using a different Java. Yeah, and then it tells me how to run the software. So in our case, we have single end reads. SE stands for single end. PE stands for paired end, in case you have paired end sequencing. And then you can just ask for the software version, number of threads. Just one is good. FRED 33 is the normal one. I think it should be the default probably in Trim-O-Matic. And then, so there is input file, output file. So you just give it whichever files you want as input and outputs. And then, I'm not sure, I can't read what's there. Oh, trimmer. Trimmer 1 is just like the different options that you may specify the ways you want the software to trim the reads. And where's the full help? For some reason I don't remember what the full help is. Quiet input files. Yeah, I don't remember what the full help is. So we're not going to actually run the software because we're really worried about time. But basically, just have to go through the help. I think there should be online, there should be a manual. And then you download the PDF. And yeah, so the command should look like this. Whoa, that's a bit too big. So yeah, something like this. This is all one command. So you have this Trim-O-Matic dash, this Java -jar Trim-O-Matic. And then in this case, it's paired end. Is there another one? It's just one command. And oh, the paired end version is more, okay, single end is here. So single end, and then FRED 33, and then just input FastQ file. So this .fastq and .fq is the same thing, and output .fastq, and then remove TrueSeq adapters, so clip adapters, and then remove leading bases based on some quality and trailing bases based on some quality, and then clip the read based on some base quality, and you can just like go to the manual and it's going to tell you exactly how to specify these. I'll try to provide I'll try to update, since we didn't really go over it, try to provide an updated PDF for the PowerPoint. And then minimum length is one that's typically used. And it's just going to give you this output file with all these filters applied. So all the bad reads will have been removed, and then reads that totally drop in quality after a few bases are also going to have been removed. Okay. Yeah, so that's what I wanted to say for processing FASTQ files. Do you have any questions before we switch to a different topic? No? Questions, FASTQ files, and reads and just raw libraries? Okay, so let's switch to screen sharing is building. Why? Resume share. Oh, that's because I was on Firefox, yeah. So sorry for the, I'm just going to show them what I meant. Yeah, I think it's running. Yes. So this, I was on the, just for the online students, I was on the website for Trimomatic, and then we're looking at the single end example on the website. So we're switching to, where is it? Oh, I need to stop this one. Okay, so yeah, let's switch to reference genome and alignments. So basically what we want is to align our reads to the genome is what's next. And just first I wanted to kind of go over what a reference genome was, just because it's not on the practical side, I mean, because it's not obvious. So mostly I would recommend working with Ensembl for reference genome files. So here's just the Ensembl website, which you might have seen, and then there's we can just click on mice. Ensembl is good because I'm not sure if you've heard about for mice and humans, the main kind of annotation set is a GenCode one. And it's actually maintained partly by Ensembl, and then can interact with a lot of different metadata databases, where you can just cross it with all sorts of other types of biological data very easily. It just works well. So if we go to the Ensembl website, just click on mouse, then we'll arrive to this page for the mouse genome. And so we can first we can just go online and just browse online on the Ensembl viewer. And so just can enter a gene name. So here I actually just entered the gene RNR2E3, which is one of the targets of this transcription factor that the source study we're working with the data of is looking at. And then you get on this page, and then you have different information. You have where's my mouse? You have the gene name here. You have the Ensembl ID, and then here you have the UniProt ID for the protein. And usually that I'm mentioning this one because you usually have a lot of information on what the gene does. It's much more functional than a genome browser. So it's really a good one to just look at. And then if you click on go to region in detail here, then you arrive to, what is this not? Oh, I didn't include the screenshot in here. Anyway, but so you have this small plot of the region here when you look at the gene itself. But then you can just go, as in a regular browser, you can just go to the actual region on the chromosome, and then you can just add different properties you are interested in, like see exons, see annotation, I don't know, methylation, documented methylation for human or mice, or any kind of what are the surrounding genes, any kind of information you might be interested in. So going back to this, the main page for the mouse genome and Ensembl, there are two sections that we are going to be interested in today, and it's actually the genome assembly section, and then the gene annotation one. So here is the search bar, right? And we have these two sections. So this is, and really what I mean is that the two fundamental things about the reference genomes are these two separate concepts of a genome assembly and then of gene annotation on top of this genome assembly. So we're going to have, so the sequence on one side, that's going to be one FASTA file. And then we're going to have everything that tells us where the genes are and just gives us the names of the genes and the different transcript that exists for the genes, and then which part of the exon are coding or not. And that's going to be the annotation file in GTF or GFF3 format, okay? One note is, so if we follow this link here, which I'm not going to do now because we don't have time, but I invite you to do so. Just, what I mean by follow this link is actually on both in this section, you just have a download the file button. I might just do it so that you, Firefox, just, okay, so I click on mouse and I arrive on this page that we've just seen. And then this is download GTF and this is download FASTA. And if you click download GTF, you arrive to a page like this, and this is your file, okay? This one there that doesn't have any weird extensions. This is a file. So same thing, you can click on the file, right click on the file, copy link location, get it from the cluster, okay? Or just download it to your own computer. And same thing for the FASTA file, but the FASTA file is going to be a bit more, this is what I meant, okay? If you click on download FASTA, you arrive on this, and you can download any of the chromosomes, and it's all like there's massive amount of files. And the one you want is actually that one, the one with DNA, soft repeat mask, and then primary assembly. Okay, let's go back. Okay, so yeah, this is a file to look for the genome itself. Okay, so this is what I just showed. This is what the FTP looked like. So the FASTA files are a bit like the FastQ files, except instead of 4 lines, you have 2 lines. For each, you have a number of reports in the FASTA file, number of sequences, right? They're not reads typically. You have a number of sequences. In the case of reference genomes, that's going to be chromosomes or scaffolds. And then you have this greater than sign, and then that's the beginning of the ID line. And then you have the ID, so chromosome 1 or chromosome 10. And then you have some annotation that relate to the chromosome itself. In this case, it's like mostly things that are specific to Ensembl. And then you have the sequence. And then just a new greater than sign, and then a different ID, and then the sequence for that other sequence. Okay, so very simple files. And then the second type of file, so for gene annotations of GTF files. They're also, originally they were called GFF2 files, and then they're basically the same as GFF3 files, except GFF3 files have been improved, and mostly for GFF3 files, what's going to happen is that they're easier, they've been structured more, they're easier to parse for a number of advanced annotation tools. But they look the same. They both look like this. The metadata is going to be the one section that's going to be different between the 2 formats. So the first, you have a series of tab delimited columns, and then the first one's a chromosome. So for now, we're just looking at things that are chromosome 1. And then the second column, I think, is the source of the annotation. And then the third column is the feature type. So we have things like gene, and then there's a transcript of that gene, and then there's an exon, and a coding sequence that overlap the exons, of course. And then the start codon is actually at the beginning of the CDS. And then there is another exon, other exons, and then the stop codon at the end of the CDS, and then other parts of the exons are actually, in this case, the exons are annotated with UTRs, right? If you're working with non-models, it's not always going to happen. In mice and human, hopefully it will. And then there's this is just where the gene and the different exon are. So that's basically the position, the start and end position on chromosome 1. So if we look at the start codon, for instance, It's from something 503 to something 505, so that's over 3 bases, which is what we expect from the start codon. And then we have the strand the gene is on, and then some metadata, so just the gene Ensembl ID in this case, and then the gene name. And so for the transcript, for instance, it's going to tell you which gene the transcript belongs to, and then it's going to have a transcript ID, etc. Okay, so this is basically what a reference genome is. You just have this FASTQ file that just gives us a sequence. You have this feature file, GTF or GFS3, that gives you where the genes are and everything. And those are the two files you want to work with when you manipulate a reference genome. GTF, so I mentioned there are 2 formats for feature files, GTF and GFS3. Really, there's the same thing. Ensembl, you can download both of them from the FTP. Just generally, I just download the 2 of them, and some programs prefer one format, some programs prefer the other format. So just give the file contain the same information. So just give each program the file type they prefer. That's going to make things more simple. Yes, do you have questions on this? No? Okay, so we're going to use this. I don't think we're actually going to use this today because I'm going to give you the simpler way. So we're not going to fully use it, but we could use it if you have time, if you want to. I'll show you. The other thing is, so basically we need these gene models to align. We have our reads and then we're trying to match them to the genome. So we definitely need the genome sequence to know where read goes where. But because of the splice junctions, we also need to know where the exon intron boundaries are. That's why we need both of these files, right? So when we align, we're going to get this. Basically, this is endpoint. So this is a goal with these 2 files. And then the endpoint is going to be alignment files. And over the genome, I'm going to show you in a second what it's going to look like. And basically, the alignment file, they're going to be in this format called SAM or BAM, and it's really the same format. So this format called SAM, and it's been defined. I mean, it's a standard format. It's been defined by not sure if it's a consortium. I think it was by the Broad. So it's basically the standard format for read alignments, and the BAM format is a binary format, so it means you can't just open it in a text editor. It's just not going to work. And they're much smaller than the SAM file because the SAM files are not compressed, and the BAM files there are a lot of things you can do with a BAM file that you can't with a SAM file. And SAM files are more for just human manipulation, but you can really want to always keep your files as BAM as much as you can. So mostly we're using BAM files, and then we're using software to manipulate them, especially in SAM tools. We're going to see in a second how to do that. And whether it's SAM files or BAM files, the file is going to have 2 sections, a header, where there's going to be some information about which genome was produced to create the alignments, and then maybe the sample that is being aligned, and other information about which command has been, which parameters have been used for the alignments, this sort of things. And then there is the alignment sections, so which is going to be a list of tab delimited lines, so which look like this. And just going back to like this. So this is our alignment section in our SAM files, and this is just the read name, and then there is a flag. I'm going to come back to that. It's kind of I'll just come back to that. There is just a chromosome where the read is mapping, and then where the alignment starts. And then there's a description of the alignment itself. We're going to just show something very quickly. And then just the sequence and the qualities of the read, which are the same one as in the FastQ. It just copies the sequence and the qualities from the FastQs into the BAM file. So this is from the specification PDF. If you Google SAM format, you're going to end up on the SAM website, that one. And there's a PDF that just explains everything you might want to know if you about the format. So the most important part is actually the alignments. I'm not sure why this red box is there. It should be just one line up. So the cigar string is the description of the alignment itself. And so one of these, the first one is 63M3I, and then 34M, right? That means a specific alignment. And then the second one is 100M, meaning 100 matches. So this is each letter. This is from the PDF from the standard. Just each letter has a different meaning. You can code different alignment events with these letters. And so 100M is just going to be an alignment between the read and the genome. In this case, a 100 base pair read without any indels. And then this 63, 3I, 34M, that actually means you have in the read alignment, it starts with 63 matches, meaning without indels, you just align every single bases 2 by 2 without stopping. And then 3 nucleotides that are only in the reference in the read. So that's a 3 base pair insertion, and then 34 matching nucleotides. So basically this is what it is. This is what this cigar string means. It means that when you take this sequence to align it to this sequence, you just start wherever the start position was for the record on the line for the SAM in the SAM file. So there's this other field that tells you where the alignment starts exactly in the genome, and then you follow the cigar string. So you start here, and then you have 63 matching nucleotides, so without indels at all. Some of the nucleotides might be different themselves, but you don't need to know that. Once you know where the read is in the genome, you know you can reconstruct the information. Then you have these 3 nucleotides that are only in the read, and then you keep going with 34 more nucleotides that just match with the reference. Basically, this is how alignments are written down in SAM and BAM files. Right. Is this clear how you can encode an alignment? And so basically, when you try visualizing that, I'm just going to Okay, I'll just go where I'm going to go back to the SAM tools program later. So basically, it is we can visualize these alignments directly on the genome. So you have your chromosomes, and then you want to see the alignments. So you can do this with one of the options, IGV, the Integrative Genomics Viewer, and basically just a software that lets you run a genome browser on your own laptop. So this is a screenshot I took on my laptop. So this is this gene that so I'm on chromosome 9 in the mouse genome. And I'm looking at this gene, NR2E3 that we mentioned before, that's related to this NRL gene that they're partially interested in. It's one of the targets. And then for this P10-REP1 sample, and I can just load the FASTA file into IGV, and then load the GTF file into IGV, and then I have my BAM file for my sample, and then I load that as well. And so there's background, which is the chromosomes, which is given by the BAM files, and then this is an R2E3 gene with its exons, and that's given by the GTF file. And all of these gray boxes are the individual reads, with the blue bars being where the reads are spliced from one exon to another. Okay, so basically that's how the software reconstructs this information about BAM files. So there's just BAM files encode these alignment information, and the software is able to make something out of it. Okay. And the main goal of all this is to be able, when you have something in your data that looks weird at the end of your analysis, you're always able to just look at this intermediate step of alignments and look if things make sense, at least up to this point. Like if there's this one gene that you thought was going to be super expressed and then you don't see it expressed, then you can open the alignments, open the genome at this position and see that whether you actually see anything aligning to that region in the genome. Right? So it's just basically one key point is that you just can't just run all the steps of the analysis without stopping and without making checks. You really need to have in mind a mean to just backpedal and check that one specific step went well. Right? So if you have a doubt at a later step, it might be that an earlier step, any of the earlier steps could have gone wrong. And this is one way to check that your alignments kind of match what you're expecting to see. Okay. Yes. So yeah, this was the exercise for this is the last slide. Yeah, we have some time to do it. This was the exercise I had for the second part of today. So basically downloading IGV yourself and installing it on your laptop this time. And then open the region at the NR2 E3. And then we're going to manipulate this one BAM file bit with SAM tools, just to use SAM tools. And then download the BAM file and its index. We're going to go over that as we do it. And then just reproduce, just visualize the data, the alignment data. So the alignment, I thought it was better for you to just visualize what the alignments were, and then we can produce our own alignment tomorrow and produce accounts as well. Just so this is one of the BAM files I have produced from this data, and tomorrow we're going to. This is just one of the files. So SAM flags, I'm not really going to go over them. It was mostly for reference, these slides. The main thing is you can't really make anything out of them directly. You have to use this SAM tool flags command to just see. That tells you basically if a read is the first in a pair or the second in a pair, or whether the liner thought that the two reads were properly paired in the genome, like aligned to the same area. So the last thing I wanted to go over was SAM tools program, which is the main one, because these BAM files are binary files. So pretty much you can't really do anything out of them yourself. You need a program to help you do that. So the overall usage is just SAM tools and then the name of the subprogram. If you just do type subtools itself, it's just going to list every subprogram it has, and then whichever options the subprogram needs. And the main subprograms are going to be view with different options. This is more for reference. And then sort, which is going to sort your BAM files by typically, the main sorting order is by chromosome and then by coordinate. So all the reads from chromosome 1 are going to come first, and then reads for chromosome 2 or 10 are going to come first. And then chromosome 21 and etc. Yes, index is to create an index from the BAM file. So which is basically an index is just a file that tells you where in the BAM file chromosome 1, the alignment to chromosome 1 start, where alignment to chromosome 2 starts, where alignment to chromosome 3 starts, etc. So that any program, outside program like IGV, can just go directly in the BAM file and load only the information that's needed, which is something that's only possible with binary files. FASTA index, same thing, but for FASTA files. And then flags, we just went over it. And then flagstat just gives some statistics about BAM files. But really the main one is view, and that lets you especially convert a BAM file to a SAM version. So one that you can actually visualize, for instance. with less or another program. Okay. Let's try to visualize the alignments. So you want to install IGV on your laptop. Should be, so just go to the website, and then, which is here. And then there's going to be a version for Windows and a version for Mac. And it should install just fine. And once you've done that, just go into the options of the program. There's going to be a select genome from the server. It has a number of genomes in memory that are available from the server. And you can just go to the mouse genome, go to the MM10 genome, which is actually a genome from the Broad. You could also create a genome yourself from the FASTA and GTF files from Ensembl to have the whichever specific version of the genome you want to use. But we're not going to do that. You're welcome to do it. I think it's the best way, but it's going to take more time. And yes, once you have loaded the mouse genome, you can just go to enter our gene in our NR2E3 gene in the search bar, and it's going to take you there on chromosome 9. And then you need to have the BAM file ready to load into the program. So then we'll go to the cluster. You're going to index the BAM file, and I would encourage you to just manipulate the BAM file a bit, open it with SAMtools view, and then download it to your laptop, load it to IGV, and you're going to be able to see the reads directly. Okay? Questions? Or, yes?
Can you just demonstrate?
For the install, or how do I compare? Yeah. Which part?
Just the size of the genome.
Which?
Because we only have 30 minutes left.
Yes.
Yes.
So I mean, this is.
I think that you open your IGV and to put the BAM files and how you create the BAI index and put BAI also, and then like
Yes.
the alignments inside the IGV.
Okay, so I can demonstrate how it's done. So I just opened IGV on my computer. I'm going to put it on the. Once it's set up, it's very simple. So this is basically what IGV looks like when you open it. And then I can go to genomes and then load genome from server. And then there's a bunch of different species. Where is musculus? Oh, it's there. Where's my mus? There it is. So I can just click musculus, and then NR2E3. Okay, and I'm at the gene, and then I need a BAM file to load. And if I go to my, oh, I'm not going to do it like this. No, I need to change the sharing. And in my case, just in my desktop on my laptop, I have already downloaded that BAM file, and we can go over what it looks. So file, load from file, and then yeah, this is just my home directory. And I've got the BAM file for this sample, and then I've got an index file going with it, and I just open it, and I get my alignments, right? And so I can zoom in on some exons, for instance. I can zoom in, and I see the individual reads, like this read that has been spliced over 2 exons, for instance. And then it tells me anything. And one thing especially is like this GAs. I mean, you can, there's a number of things, and depending on what you're interested in, if you're interested in like allele specific expression, for instance, looking at transcript for 1 or the 2 parental alleles, you might be interested in reads that have a different genotype. than the reference. In this case, it's also going to show that, like whether like this one read for some reason has 2 nucleotides that differ from the reference. I'm not sure exactly what so these are the reads, the nucleotides in the read. I'm not sure if I can. No, and then, so this read has GA. I'm not sure exactly what's in the same column. Yeah, you can check that. But the main thing is typically checking that you actually have, generally the main, and then there are different, yeah, squished, we'll plot it like this. And this is basically the expression of the NR2E3 gene. Right? And we could compare it with the same region, but in one of the sample that's knocked out. And then we should see that this other sample, because there's no NRL to activate this one downstream gene, then we should see the expression is going to be much lower, which we should also see in our differential expression analysis principle. So if you have weird results in the expression analysis that you really don't understand, you can always go back to the alignments and just check that things are the way you expect them already at the alignment section. Yes, and then maybe the other thing I can show is SAM tools on the clusters module. So just going back to the cluster. And if I do scratch, okay, I'm here. And if I'm copying this to the demo directory, so I have this one file, one BAM file. And then if I try to head that file, just print the first 10 lines of the file. This is, I mentioned, a binary file. So it's going to look like this. These are the first 10 lines of the file. It's just, a binary file doesn't have lines, so none of this makes sense. And that's basically whenever you see this sort of result, it means you've printed a binary file directly to the terminal. So what you should do instead is convert the BAM, if you want to view it, not if you want to give it to a software, you're going to give a BAM file directly, and the software in principle is going to be able to handle it. So what you want to do is load the SAM tools module, and then use the SAM tools. If I do just SAM tools like this, as I mentioned, it's just printing every single subcommand. So this is just basically what I was talking about. And so it's printing every single subcommand it knows. And the main one is a view command, which is this one. It says conversion, which is, and the name of the command always never really made sense to me, but it is the view command. And then if you just do SAM tool view like this without anything, it's going to list all the possible options you have for that command. And then the H option converts it to a full SAM format. So it's a SAM format with the header. The default is to just behead the SAM and just remove the header, show the records directly. And the sample was P10, I think, like this. Yes. And if I send this to less, so just, it's actually, this time if I do head, it's not going to print just random characters like the previous time. It's actually going to print text. Right? So this is the beginning of the file. And this is all part of the header with this @ signs. So it starts by saying the version of the BAM file, the version of the BAM format or SAM format that's used, and then the file is sorted, in this case, by coordinate, as I mentioned. And then these are all the chromosomes. So actually the chromosome order in this case is 1 and then 10 because it's alphabetical. And this is the length of chromosome 1, just like so that the BAM file already encodes a number of information about the genome, especially in case you're trying to use a BAM file as a genome that is not the same version of the assembly, which would not make sense. So if I use less instead of head, and then, so this is the same beginning, and then I can scroll down, and then I'm going to go over I'm not sure how many chromosomes mice have, but I guess 20 chromosomes. So 20 chromosomes plus a number of scaffolds that have not been placed anywhere. And then some information about the star command that was used to create the file, and then the alignments themselves. So if I go right, then I can see I have my sequences, and then a number of additional fields that we're not concerned about. But basically, this is one read name, and then this one is actually a reverse read. Oh, it's mapped to the negative strand without indels. Yes, basically that's a basic way to use SAM files, BAM files, and SAM files. And so these are the couple options I had mentioned in on the PowerPoint. The dash capital H just prints the header. So I route to the end of the header, and that's the end of the input for less. So this command here did not print the alignments at all. And then without any of the two H options, then I get the records alignments directly. Yes. Yeah. Play with some tools, and then try to install IGV on your laptop. It's always going to be useful. Yeah. Yes. Oh, index file. You have to create an index first. Oh, it should be.
I don't have this. I don't see. Yeah,
Yeah, basically, when you need to use some tool to index the BAM file. You can either do it on the cluster, or if you have some tooling from your laptop. Okay. If you go to the end. So the easiest way is just to create the index on the cluster where some tools will be installed, and then download the two files together, and it's great. So it's some function of this. Need a S. No, S. But you need to install something on the laptop. It's like something that's going to be like but S. No, you can't S on the cluster. You need to always the recipes will always be found from the laptop. But so you're on Mac, so you can just do it if you or another terminal that's going to be running your laptop. Oh, you've done it already. Just open the terminal. Second video. And then, so this one is on your laptop. This one is this, and this is, and for so this one is actually running this shell here. It's running on the culture somewhere. This one's running here. Yeah, and this is where you want to do this. As in the yes, once you likewise and then column, and then column. So and after so this is what this is. So it has been changed to where where the file, and this address has to be the file you want to transfer. Or the directory the file you want to transfer. Then you can just drag it from here to there, or just maybe there's buttons where you just transfer. There's not usually but there's usually a button to transfer things from that. But this so if you Control C, that's yeah, this so this is a directory. You enter this on the bar. So just copy that location.
So you have to reset your folder from the sequence, and then it would be yes, then those that you use.
That's something she has. That has to do with the way and it's like each control should I don't know, there's I start where you so yeah, so
So you will online.
yeah,
So after you do this, then
so
you run the sound because then you have to count
Yeah. But I'm not able to generate how.
the alignment and how
If I'm using server, it is not augment server. Augment server will install the whole So I'm working on the server. So how do we hold this
So you will have to
okay.
but you will for that. So you can continue from later on. Maybe you know.
Yeah, tomorrow I will go to prepare one, not all the samples, only one sample for the time.
Yes, yes,
And then try
yes,
to, okay.
yes. Do it like this. This is how this workshop. How we do.
Yeah, I know some of the basics of the difference, right? I use this video. Yeah, and I also use the D6. But there is a okay, but there is a difference in the differentiating expression. In these, yeah, in these two I'm getting 40 differential. Yeah. Which are getting 100. Yeah, in this they use it.
So this is but it's very hard to switch. So this is
Yeah, in this package they use three.
Okay.
And question then I think okay. My name they're also
And you mentioned tomorrow we'll talk about
So you
but human genome.
but you should and after alignment also you know that once the is
okay. So after alignment.
but then so you have to that's why you have a GPR.
Okay.
Yes, for the annotation. So after annotation, and then
Okay, align with those.
this is here what is
And then
Okay.
they
Do
generate.
you know how to do this? Align
if you have like specific questions.
Okay, because I have two
one is
now three suffers and mouse it's a paired, but humans is a single line. So I want to generate countable for both.
Yes. So you use the this is clear.
If I will get countable, then I will analyze. So my point of this workshop is to how to align the
So tomorrow is the
okay.
we will be just only alignment.
this path is this how this is what here the cluster. So the address of the cluster
理解上去看，我就是说可以跟着对，
I guess it's already installed, but it's just. Yeah, just like wondering, should be seen so on which. Thank you.
Also, questions online?
Today you had a lot.
Okay.
It was simple.
For
For
like,
example, if I need to, yes, if I need to, like, for example, copy, the folder to my scratch folder, or which command line to CP, which command line I use, like, so I example. It was 21 people and 8 online. 29.
What happened? Oh, so you're what happened next to I'm confused.
No, it's because you are done, so
So I think I can record from mine.
oh, you could?
I don't know if try it out tomorrow, but I don't have to record, but I'm record. I think they just want to record me, but not you. So I'm gonna play with it tomorrow. As a case I was like, so you guys see the B part, I just want to record from my end. But we'll find out. So yeah. Tomorrow, let's say.
So today we come. Letting the students do it. So he's just going through asking for everything. And because he's doing a GB and 5. And he's doing star and high sat. Both cases. And then he's doing stuff as well. And then he's also doing counts with stars.
I guess somebody forgot their makeups and here tomorrow.
Should I stop the recording?
Yeah.
Do we still have students online? Do we still have
You
people?
can end the meeting now.
Maybe check. How do I make this bigger?
Yeah, I was asking, I can ask you if you guys want to take 2 hours or something. I figured you guys are
Oh, chat. I'm just going to stop the recording, but not end the meeting.
Yeah, so that way